Welcome back. Now that we have the basics down, we're going to get into the versions of the immunity. We're going to be talking about T cells here, and then later on we'll get into the B cells. So our T cells are going to have three distinct life stages that occur in three different places in the body. They're going to be born in the red bone marrow, they're going to go to school in the thymus, and they're going to be then deployed out to work in the lymphatic organs. So we've already talked about the formation of blood cells in the bone marrow, so we're going to skip right to their schooling. And we're going to go to the University of the Thymus here. So when your T cells arrive at the thymus, this occurs during fetal development, there are hormones called thymosins that are going to stimulate the T cells to develop a surface antigen receptor. Their entire job at the thymus, when they are at the University of the Thymus, is to develop this receptor. What the receptor looks like is based on the specific set of genes that particular T cell got. And the best way I can describe this is picture that our alphabet only has 26 letters, but we can rearrange them in a huge number of ways to get totally different words. So you have specific cassettes of genes that get allotted into these different populations of immune cells, and it allows for pretty much almost theoretically unlimited versions of these receptors. And each one is going to be a lock and key with the antigen it would bind to. So each one of these receptors is slightly different in its shape. Once the T cells have done that, then they are capable of recognizing their antigen if presented to them by an APC. That means they are what are called immunocompetent. Once they get to this point, now it's time to test them. So they're going to be administered a final exam, and the cells that are going to administer the exam are a set of self cells called the reticuloendothelial cells, or RE. Think of them, I guess, as the teacher. They're giving the exam. What they're going to do is they are going to present self antigens to the T cells. There's going to be two ways the T cells can fail the test, and it's going to be presented to them sequentially. So we're going to start with what's called positive selection. If the T cells were incapable of recognizing that the RE cells are self, then they fail the test. So one of the first things that an immune cell needs to be able to do is tell self, particularly when it comes to the MHC antigens. So these are the major histocompatibility complex molecules that I was talking about before. I said these are the primary self antigens. You have to recognize your self antigens. So if you don't, and you can't recognize self, then you're not going to know what foreign is. So they failed the test. So if you fail at positive selection, you don't even get a chance at negative selection. You don't even get a chance to move on to the second round of the test. If you recognize the MHC, you pass positive selection and you move on to negative selection, which would be now we're going to look at the actual self antigen. We're going to look at the hot dog in the hot dog bun. It's a self antigen, so the T cell should leave it alone. If it attacks it, then you failed the test, because that means you would attack your own tissues. So let's take a look at this concept of the test here a little bit closer. So what we're seeing here is the purple cell is the RE cell. Okay, so this is the purple cell, and this is the developing T cell that is being tested. Now, This piece right here, okay, ignore this piece, the self-antigen, but this receptor on the surface that looks like this, that is self-MHC. That is your hot dog bun. The self-antigen is the hot dog. So when we say that the T cell has to recognize the MHC, what we mean is it has to be able to line up with it. And so you can see that this one with this point right here is absolutely not going to be able to line up. But this one can line up. Now remember that this receptor is generated randomly based on the set of genes this T cell was allotted during development. So it's not as if they didn't study for the test or something like that. It's completely random which ones will actually fit with our self-MHC. So these guys failed the test. They cannot recognize the self-MHC, which means they're not going to know if it's not us. These guys, they passed the test and are now going to move on to negative selection. Now we're done dealing with the hot dog bun. We want to look at the hot dog. This is a self-antigen that is being presented, which means that the T cell should look at it and then go, whoa, this is self. Let me back up. So if it reacts by binding tightly to the antigen, it just failed the test. Okay. If it looks at the antigen but does not react, it does not bind tightly, it has passed. If you fail this test in either one, the cell line dies via apoptosis. So that's a pretty significant result of failing a test. If you fail the test, you die. If you pass the test, you get to live. So it's only this population, cells that can both recognize our very specific MHC and also not react to the self-antigen, that pass the test. It's about 2% of them that pass the test. So there's a 98% fail rate when it comes to the testing in the thymus. So the University of the Thymus is not kidding around. And that very high rate of failure is because this is completely random. It's completely random what genes are allotted out to each cell, and some of them will fit and some of them will not. Most of them will not, in fact. Once they've passed, they are then going to get sent on to our lymphatic tissues and the organs. So they're going to go to the spleen and they're going to go to the lymph nodes and all of those follicles we talked about. And then they're going to hang out and wait until they get to interact with something. So let's talk about the 4 classes here that we're going to see in the T cell line. The true effector of cellular immunity are the cytotoxic T cells, and here are all the other names for them, okay? They are the ones that walk up to another cell, punch it in the face, and kill it, okay? They deliver what's called a lethal hit to whatever the target cell is. Helper T cells are going to be the boss of all of this. These are the cells that everybody's running up to going, hey, T cell, T cell, T cell, I ate this thing. Can I go ahead and eat more of it? I want to make some antibodies. Can I do this? Everything that happens in the immune system requires the okay of the helper T. Regulatory T cells inhibit the immune system. So they are going to just kind of pump the brakes. Make sure that while we begin these processes and we're releasing all of these cytokines and we're going through massive rounds of mitosis, that we don't go too far, because that is just as bad as not responding. And lastly, we got the memory cells. Because we're in the 3rd line, we're always making memory. Okay, so when we talk about this true cell-mediated immunity, We are talking about the killer T cells, the ones that punch things in the face. So this is a direct attack. The cytotoxic or killer T cell is going to interact with either a foreign cell or a host cell that has become diseased, infected, has cancer, any of those. Because we're in the third line, we're going to remember what that antigen is, and we're going to be able to kill it a lot faster in the future. But because we are dealing with specific immunity, we do require this antigen presentation to occur. We've got to get the okay from a helper T before we let loose all of the cytotoxic T cells. So let's talk about that. So we saw this before. The APC encounters an antigen, processes it, pops it back out onto its MHC hot dog bun on the surface, and goes and finds T cells. Now, what we haven't talked about yet is the fact that there are actually two different types of MHC. One is going to speak directly to cytotoxic T cells. Two is going to speak directly to helper T cells. So we're going to talk about this in a second. Once we have responded and we've decided that we have the correct epitope, we're going to go through a co-stimulation, kind of think of it as a double handshake. So the first interaction is with the MHC and epitope, and then we kind of have a double handshake. It's kind of like you got to know the handshake, you got to have the secret word, you got to know what the passcode is, because we want to make sure that we're doing the right thing. And so there are layers of protection here. Once that happens, we then go into clonal selection, which are the rounds and rounds and rounds of mitosis that are creating clones of this authorized cell. Some of them will be the effector cells that are going to go deal with this epitope right now, and others are going to be the memory cells that are going to sit back and wait until you see it again. So let's talk MHC for a second. So class 1 and class 2, we're going to do a comparison here. MHC1 is found on all cells that carry a nucleus. So any cell that has a nucleus is going to have MHC1 on its surface. Class 2 is only found on APCs. Now, APCs, a dendritic cell, a macrophage, a B cell, all have a nucleus. Therefore, they also have class 1. But they're the only cells that have class 2. Cytotoxic T cells recognize MHC1. Helper T cells recognize MHC2. Now, going back to what we've talked about, we know that we have intracellular pathogens that are going to be targeted by cytotoxic T cells. Therefore, the antigens that would normally be displayed in class 1 are generally endogenous or intracellular because they are asking the T cell, this cytotoxic T cell, to do something about it. The exogenous or extracellular pathogens are what are going to be displayed on your class 2. So, for example, the B cell engulfs a bacteria and then is going to present it on its surface to a helper T cell to ask the helper T cell, hey, can I make antibodies against this? Okay, so we're still looking at that concept of intracellular versus extracellular. Now, this is an important line, okay? This is why I have taken this table from another book, because it's a really good way to look at this. So what we're looking at is what's the message, okay? So when I have an intracellular foreign antigen on MHC1 and it's an APC, okay? So it's an APC, because remember that APCs have both 1 and 2. So here we have an APC And what it's saying to the cytotoxic T cell is, hi, I belong to self, but I've captured this foreign invader. This is what it looks like. You need to go kill any cell that shows this. So let's say it's a virus, and we've got viruses going into cells. What this is saying is, hey, I need you to go find anything that has this, and whatever cell it is, kill it. Now, in order to do that, a helper T would have had to give the okay, but we won't get that deep into this. If it's a cell that is not an APC, so any other body cell, what it's saying is, hi, I belong to self, but I have been invaded or I have become cancerous. You need to kill me. So the difference in these two populations of cells is that if an APC is displaying it, it's saying, hey, cytotoxic T cell, I okayed this with a helper T. Go find everything that looks like this and kill it. All the cells that look like this, kill it. If it's a body cell that is not an APC, it's saying, I need you to kill me. I've been infected. I have a problem. I'm turning cancerous. I need you to kill me. So very different message there. When an APC is showing an extracellular pathogen to a helper T, the message is, hi, I belong to self. I've captured this foreign invader, and this is what it looks like. Help me do something about it. So if it's a macrophage, this is where the macrophage is going, hey, I have eaten this, and it is so good. So is it okay if I go eat all the other things that look like it? If it's a B cell, it's going, T cell, T cell, T cell. Oh my God, I have been waiting my whole life to find this thing right here, and I am the only cell that can make antibodies against it. Can I go make antibodies? So it's asking for permission. This is what we've talked about before when we were talking about APCs and what are they asking for? Okay. So differences between the class 1 and class 2 here. So what happens when the cytotoxic T cell has been given the okay? This is going to be the third example of our immune system functioning by poking holes in things. So this is very similar to the natural killer cell in that we are going to use perforin proteins, and then we're going to throw granzymes into the target cell, which is then going to cause apoptosis. So now you might be wondering why we have two populations of cells that appear to do exactly the same thing. The difference is that natural killer cells are innate. They're in the second line of defense. Cytotoxic T cells are adaptive. They're in the third line. The third line has memory. Natural killer cells do not have to go ask permission from a helper T. They just look for anything that looks a little bit weird and they kill it. They don't ask any questions. They don't ask permission. Cytotoxic T cells have to go through layers of permission before they get the okay from the helper T. That's because they have memory. If a natural killer cell kills off a couple body cells that it wasn't supposed to, it's not the end of the world. It's not going to remember that particular antigen and make it its life's business to kill everything that looks like it. But the cytotoxic T cell with the memory cells absolutely does that. It remembers the antigen and says, this is now my one job in life, is to kill everything that looks like this. So we have to be very, very careful with the cytotoxic T cells. We have to make very, very sure that the antigen is not a self-antigen. Whereas over on the natural killer side, if they kill off a few of our cells, it's not the end of the world. There's no memory. So that's the difference between these two populations of cells that appear to work exactly the same. Now, the helper T cells, I've been saying, it's the boss, it's the boss. Nothing happens without a helper T. And here you go. Pretty much every piece of your immune system outside of intact skin and mucous membranes either requires or functions better with a helper T. So over here, these green cytokines coming off of these actually help out the nonspecific, the second lines. Now, do you absolutely have to have the helper T for the second line to work? No, but everything works a lot better, okay? The pink line is your humoral immunity. This is when we're saying, hey, B cell, it's okay to go do what you want. Go make those antibodies, which we're going to talk about next. But it requires, this is a requirement. You got to have the helper T. And over here is what we just looked at. This is cellular immunity. You can't get all of those cytotoxic T cells without the okay from the helper T. So when we say that this is truly the cell that runs the immune system, it is absolutely true. When we get into disorders in a little bit, we're going to talk about HIV. And one of the reasons that HIV is so devastating is that its cellular target is the helper T. It effectively knocks out nonspecific defenses, humoral immunity, and cellular immunity because it targets the gateway cell into the immune system. Pretty much everything requires this helper T cell.